Can you hear me? Okay, there we go. All right. Okay, so now we get to give you bird and tree people a break. We get to talk about coral restoration. Yay! All right, so I'm actually really excited about this panel. We have Dr. Lyza Johnston, who's going to come up and talk about NGO-driven coral reef restoration in the Commonwealth of the Northern Marianas, status, and next steps. And you'll be seeing her frequently at this panel, and it's always a joy to have her talk to us. So if we can we please welcome Dr. Lyza?
Hold it. All right, hello. Ooh, that's loud. Thank you all for being here today. As Marie said, my name is Lyza Johnston, and I run an independent coral conservation and research organization on Saipan called Johnston Applied Marine Sciences, or JAMS. The name was really just for the acronym. And I've been on Saipan for over 10 years now, working on coral reef conservation. Unfortunately, the last 5 years have been largely focused on coral reef restoration. And so I'm going to talk to you about 4 years worth of work in 12 minutes. I have a lot of slides. I'm going to zip through them very fast, but please track me down sometime if you have any questions about anything you see here. All right. Oh, okay. So the reason we need to do restoration, unfortunately, is because coral reefs are really in crisis globally, and the CNMI has been no exception. From 2013 to 2018, we experienced 3 pretty severe thermal stress and bleaching events caused by marine heat waves. And then in 2015 and 2018, we had 2 super typhoons that came right over the islands. Based on some surveys that we did at around 35 sites on Saipan in 2018 and again in 2012 and again in 2018, we found that coral cover declined by over 66% on average, and this resulted in, obviously, losses of ecosystem services. There was changes in coral community structure. We lost about 90% of the acropora species on these shallow sites and about 75% of Pocillopora species. So these are pretty thermally sensitive species, so that wasn't too expected, but it was really a drastic decline. So as a result of this sort of mass mortality, active coral reef restoration really became a local management priority. Restoration was added to several management guiding documents, and we've also, developed a draft action plan for coral reef restoration. The major goal in this draft plan was to restore or enhance coral reefs to improve the CNMI's resistance to sea level rise and increase storm events. So again, we had bleaching events and some major storms that came through at the same time. So the importance of coral reefs as coastal protection was really in the forefront of people's minds. And so this is a major goal of our restoration efforts, and also to improve fisheries habitat. So from there, we got some initial funding from NOAA and collaborated with all the local resource management agencies, and we established the first ocean-based coral nursery in the CNMI in 2019. And we began supportive breeding efforts in 2021, and really our goal all along is to restore with resilience, to steal a phrase from the Hawaii lab, but we're trying to produce corals that are genetically diverse so that they have higher adaptive capacity, and also we're actively trying to propagate thermally tolerant corals so they can have a better chance of withstanding these heat waves in the future. All right, so it started out with figuring out where to put the nursery. This was a little harder than we expected. This is a challenge that we have in these high energy islands that they don't have in the Caribbean, in the Atlantic, where they have a huge, what is that called? Continental shelf, yes, thank you, Steve. Man, I miss Steve. Thank you. They have a huge continental shelf. It's lower energy. There's plenty of habitat. We, on the other hand, have these islands that just drop off really fast. It's very high wave energy outside of the lagoon, and so we were kind of limited to this sort of narrow section of the lagoon in the center where it gets a little deeper, and there's a little sand. So we looked at other human uses like diving and parasailing, benthic cover and habitat, depth, and then other points of interest like long-term monitoring sites, protected areas, and other points of interest like the Maritime Heritage Trail and other marine artifacts. And so we put all of that together and came up with a map. Oops. Came up with a suitability map, and where we have white is the least suitable, and this dark green color is the most suitable. And so we went out and did some, scoping and ground truthing, and ended up putting the nursery right in this little corner of the Manigaha Marine Conservation Area, which may or may not actually be within the boundaries. You can talk to Robbie about that if he wants. But so it's right here in the lagoon in this little deeper area. It's sand bottom. So we put in 10 of these tree structures to start with. All right. We originally started with 5 species of acroporid corals, 2 staghorn acroporid corals, which are branching, high structure corals that grow really fast. And we know that these corals do well on these kind of tree structures due to all of the work that's been done in the Caribbean with similar species. We also picked a couple other ones, Acropora abertinoides. This is this species. This used to be, this is a really important habitat former. It's really big and arborescent, and it used to be pretty common on the really shallow reefs of Saipan, and it was hit particularly hard during the bleaching event. So we picked those guys, and then Acropora globiceps, which is an ESA-listed threatened species, and so we're working towards the population enhancement of those guys. Again, we're trying to increase the thermal tolerance of these corals, so we collected, all of our initial parent colonies were collected in 2019 after we had the most severe bleaching events that we experienced in Saipan in 2017, and so we collected large adult colonies that were healthy, and we presumed, obviously, that they had survived the 2017 bleaching event and therefore had demonstrated some thermal tolerance already. We then tracked some growth and survival. This is just some data for the first year for our Acropora Pulchra genotypes, so the genetic individuals. And we looked at it by genotype and by collection site, and we did this for all of the different species. We looked at survival, obviously, and after the first year, we had really good survival at 99% across all of our corals. So we decided that, that location was an okay location for a coral nursery, and we would continue there and expand. So from 2021 to 2023, we added so far 13 more trees, 8 table structures like this. And we actually now have 13 species from 5 genera in the nursery. 11 of them we are propagating asexually in the nursery, and 2 of them we just have juvenile corals that we've collected babies, and I'll talk about that in a minute. We began collecting massive corals, Goniastrae and Prydes corals, and using microfragmentation techniques, which gives us these little tiny colonies, and then they grow faster, and then those microfragments from the same coral can fuse back together. So when we outplant them, they can resheet larger areas faster than they would if they were larger fragments. So we started doing that. We also started doing coral spawning operations or supportive breeding. Talk a little bit more about that. Again, so many corals reproduce sexually by broadcasting their gametes into the water colony, and this happens only over a couple of nights a year in the summertime. And so we go out and we collect these gametes, so these are eggs and sperm bundles. We collect them, we fertilize them, and we keep them in culture until they're ready to settle. And then we settle them on substrates, and we'll use those to outplant on degraded reefs. And then again, hopefully that will be increasing, obviously the number of corals, but also the genetic diversity of those corals so that they'll be better able to adapt to the changing environment in the future. We also developed some novel settlement substrates to improve restoration outcomes using these sexually propagated corals. There's actually 2 additional talks on these, so I'm just going to zoom through this pretty quick for time. Don't worry, you'll hear more about them. These are just some nice baby pictures, like our baby photo albums. These are the beads in the reef. So they, just get placed in these little nooks and crannies on the reef. We don't need any sort of adhesive. The idea is that they'll be easier to handle and disperse over really large areas of reef so that we can hopefully, maybe even without diver input. I mean, think about, just sprinkling them off the back of a boat. Okay, so these are babies. These are ones we've done some pilot outplanting, and the corals grow pretty fast, and they grow over the beads, and they attach themselves to the reef pretty quickly. So that's exactly what we were hoping to see. All right, these are just some other substrates that we've developed. We have some additional funding the next 2 years to further develop these guys. Yes, so then we had, after a couple years, we had a lot of those staghorn corals had been growing, and we've been propagating them in the nursery and creating more and more corals. So we needed to start outplanting them. And staghorn corals was a particular priority in the DFW Wildlife Action Plan for the Commonwealth of the Northern Mariana Islands. They're very important fisheries habitat in the lagoon, and again, also very important for coastal protection. And after, in the lagoon, there used to be, acres and acres of these healthy, live thickets. And by 2015, these had all died. And so now there are just these dead structures out there that will erode pretty quickly. And so we wanted to try to recolonize these thickets, and this really hasn't been done before. So we did some pilot outplanting to see if the corals would survive on these thickets. And we use structure for motion photogrammetry to create large-scale orthomosaics and models of the reef to model these corals. And we found that they actually did pretty well. This is 2 years after outplanting. These are our outplanted corals. So we think that this will be a valid method to recolonize these thickets so that we don't have to put any artificial substrates out on the reef, and we can hopefully not lose all of this existing structure that's out there. All right, I'll just go really quick. We also went through a pretty rigorous process to prioritize forereef sites, so reefs outside of the lagoon in Saipan. We collected a lot of data to sort of answer these questions, and I'm out of time, so I'm actually going to go pretty fast. We collected new data. We got some input from managers on how they thought that these sites were relevant to that coastal protection and fisheries habitat goal. And then we ended up prioritizing a few sites for restoration in the next few years. So we're going to start with these 3 sites and then move down this list a little bit, keeping in mind these scores. And we've done some pilot outplanting at a couple of these sites, but we'll be upscaling this year a lot. Oh, these are just some pictures of our pilot outplants on the forereef. They're doing really well. Very happy. Next steps, so we're going to expand the ocean nursery. We're going to go up to 40 trees and 10 tables. We are expanding our land-based facility. We just got 2 coral spawning systems from the coral spawning lab people in the UK. They essentially make these kind of modular plug-and-play systems where you can manipulate the environmental parameters to get the corals to spawn when you want them to. So we are going to start with Acropora globiceps in these systems and try to improve our sexual reproduction of those corals because they are ESA listed. So we have these now. We are working on getting them set up, but they're pretty fancy and we're pretty excited. We're also building a outside seawater system with 15 raceway tanks and some other stuff. We're going to keep collecting data on growth and survival, both on the reef and the nursery. And we're also doing some thermal stress testing using the CBASS system. And Ilan Bubb will be giving a talk about that in a minute. Again, we're going to upscale our outplanting efforts and monitor our outplanting sites. All right, this is just an idea of what we're going for. These are ACE buckets on my porch. And hopefully this year we're going to have something that looks more like this, which is the Coral Vita setup in the Bahamas. Thank you. A lot of people were involved in getting this nursery set up and running. Too many people to name, but I tried to name a few of them here. We have a lot of local and federal partners. And that's it. Time's up. Sorry.
I think probably we'll have a little bit time for questions later. I do want to make a plug for Lyza's field trip. If you haven't signed up yet, you will actually get a chance to snorkel and see the nurseries underwater. It's not scuba based, so I strongly suggest you take the opportunity to see it. I am pretty stoked about it. Okay, and next we are going to have, it's really dark here, and my glasses really need to be changed out. Ilan, sorry, Ilan Bubb, actually from Johnston Applied Marine Sciences as well. And he is going to discuss thermal tolerance among nursery Acropora Pulchra genotypes. Also, Guam people online, I see you, so feel free to ask questions as well. I'll let you adjust that to what's comfortable for you.
You know what, I'll just hold it. Okay. Okay. Hey everyone, my name is Ilan Bubb, and I'm a research associate with Johnston Applied Marine Sciences, as well as a PhD student with the University of Amsterdam. And I am very happy to be here today to present on the research that Jams and I are doing, assessing thermal tolerance across Acropora Pulchra genotypes. So I think in a room full of biologists here, I don't really need to get too far into how climate change is detrimental to all of our species of interest. But what we do know is that climate-driven warming waters are especially potent to the mortality of corals pretty much globally. And the warming of our waters has gotten so bad recently that this past summer, NOAA has had to actually create 3 new bleaching alert levels. So NOAA Coral Reef Watch, which does projections of coral bleaching events, used to have a max alert level of 2, which discussed or that represented wide-scale severe bleaching, which sounds bad enough, but now we have 3 new levels with alert level 5, meaning near complete mortality. So that's kind of what we're dealing with right now in the coral world. I'm so sorry. But yeah, so these are the 3 new bleaching levels right here, and near complete mortality, which is just really scary to think about. But as Dr. Johnston was just talking about, Saipan has already experienced some pretty severe bleaching events. We've lost about 66% of coral cover in our survey sites. And what this really goes to show is that that for restoration to be effective, we really need to take into account these extreme bleaching events that are going to be occurring more commonly going into the future. And if we do not, then we really risk wasting a bunch of time and a bunch of money and a bunch of effort. And one of the ways that we can do this is by restoring with thermally tolerant corals. And what does a thermally tolerant coral look like? Well, you might have seen images like this from National Geographic, maybe yourself if you snorkel or scuba dive, but it's this kind of classic image where you see two colonies of the same species, but one is completely bleached and the other is doing very well, is very resilient. And what we're probably looking at here in situations like this are two distinct genotypes of the same species, and one of them, for whatever mechanism, whatever reason, has been more resilient to bleaching or is able to recover more quickly. And scientists have noticed this kind of thermal tolerance variation in corals for a while and have recognized its importance for understanding the future of corals and the importance of it in terms of restoration practices. And because of that, there is a lot of research going into understand why corals of the same species demonstrate these variations in thermal tolerance. And what we've kind of come to understand is that there are three main axes of thermal tolerance. The first one is that there are environmental factors, where it's generally understood that corals that are acclimated or growing in more stressful environments, such as warmer waters or more fluctuating waters, are more resilient to heat stress than corals of the same species that grow in cooler, more stable waters. We also know there is a genetic axis where there are certain alleles in populations that allow the coral to be more resilient or more efficient in handling heat stress. And then we also know that there is a component of the symbiont itself within the coral. So the algae that lives within corals also have varying levels of thermal tolerance. And if a coral happens to host an algae that is more thermally tolerant, then the coral itself will also be more thermally tolerant. And so all three of these factors kind of come together to form this gradient in thermal tolerance for any individual coral colony. So our research is really interested in all three of these axes of thermal tolerance, but we're also interested in understanding how thermal tolerance changes across multiple species, across multiple different functional groups that we're going to be working with. And we're proposing to do a lot of work with wild corals to assess thermal tolerance in situ. But we're also planning to start with nursery corals so that we can inform on the ongoing restoration work that is happening right now. And that kind of leads us into this pilot experiment that we conducted earlier this month with two genotypes of Acropora Pulchra. So this is a very important staghorn species to the CNMI. It forms these really dense branching colonies that provide habitat for other species, but also provides ecosystem services for us, such as wave attenuation. And this was one of the species that got hit really hard by the bleaching events that were discussed a little bit earlier. And so after the bleaching in 2019, Johnston Applied Marine Sciences went out and collected 24 colonies of Pulchra across the entire lagoon. And we found that of those 24 colonies, they comprised 18 distinct genotypes. And so we conducted trials on two of these genotypes, one found in the northern part of the lagoon and one found in the southern part of the lagoon. And we chose these two species specifically just because there was a lot of biomass in the nursery for us to work with, but also because it allows us to start looking at some environmental priming that might be occurring in our lagoon. And so a lot of people think of our lagoon as being pretty homogeneous, but it turns out data from BECQ and DFW shows that there are variations in temperature profiles in different parts of it. Specifically, what we find is that while the northern and southern parts of the lagoon have a similar average temperature, we see that the southern part of the lagoon fluctuates a lot more in their highs and lows. And that could be evidence of a potential priming effect that we would be interested in. And it was also pretty cool that this data right here were taken from two loggers from BECQ that were right next to the collection sites of the two genotypes that we're working with. So it really goes to show, or it really represents what the habitat characteristics of the genotypes we're working with. But to actually measure thermal stress, we are using something, the Coral Bleaching and Automated Stress System, or CBASS for short. And this is designed to be a portable, modular, and affordable system that can be rapidly deployed in field or in lab-based settings to assess thermal tolerance in your corals of interest. And this is what our setup looks like right now. In general, you have 8 tanks. We're just seeing 4 of them right here. Each tank is independently controlled with a heater and a cooler in it. And these heaters and coolers are connected to an Arduino microcomputer that can be pre-programmed with different temperature ramps so that you can look at different thermal tolerances with your corals. This is what it looks like set up. When I'm sitting behind this table for 18 hours, this is the view that I get. You can see that there are 8 tanks, again, each one independently controlled, with light systems that mimic the light profiles that you would see in the lagoon, as well with a sump and a sump system that circulates water all the way through it. So most CBASS experiments kind of generally follow the same pattern. They take about 18 hours, and each tank is set to have a thermal profile that will cycle through. In general, tanks ramp up to their maximum temperature over a period of 3 hours to not shock the corals. They're held at their maximum temperature for 3 hours. That is mimicking the stress that they might experience in the environment. And then it ramps down over an hour where, when it finally holds for 11 hours overnight, and you come back and do some metric assessments. We decided to use pigment, the color of the coral, as our response variable to measure stress. And so to measure pigment, we took baseline photos at the very beginning of the experiment, another set of photos of our fragments at the end of the ramp down, and also at the very, very end, at the end of the 18 hours. So this is kind of what our pre-experiment setup looks like. We developed these kind of acrylic plates that allowed us to mount our coral fragments to. We went to the nursery, we fragged all the corals, mounted them, and then placed them in the nursery on these platforms so that they could heal for 10 days before starting the experiment. And this is what our experimental design looks like. We set up a baseline temperature of 29 degrees Celsius, which is taken from NOAA's Coral Reef Watch to represent the general max temperature that these corals are acclimated to on a yearly basis. And then each of these plates that we had 8 plates for 8 tanks. These were all randomly assigned one temperature treatment, so either +0, +3, +6, or +9 degrees Celsius over the baseline. And we had 2 plates for each of these temperature profiles. In addition, we put 2 fragments from each of those genotypes onto each plate and randomly distributed them. So you can see that none of them are exactly the same to help correct for any tank effects that might occur. But this experimental setup allows us to have tank replication and fragment replication for a robust experiment. Pigment analysis. So like I said, we used color to assess the stress of these corals, and that kind of looks like this. So like I said, we took pictures of all these corals at all these different fragments at 3 different time steps. And at the very end of the experiment, we brought them into Photoshop and when we took these photos, we also had these color correction cards, these white and black cards that we could stretch the image colors. When we brought them into Photoshop, we converted them to a grayscale image so that the pigments are on a scale from 0 to 100, with 0 representing absolute white, which would be the white card right there, and then 100 being absolute black, which would be the black card right there. And then in Photoshop, we were able to kind of selectively draw a polygon around the fragment, and Photoshop has an awesome tool that allows you to calculate the average pigment of it. And that kind of looks like this. So here you can see one fragment from each of the treatments over the 3 timestamps, and you can see how the color pigment changed over time. One coral fragment from each of the treatments. And you see that at max temperature, at that +9 degrees Celsius over baseline, the final corals were pretty much all bleached at that point. Important to note that our starting points were not exactly even the pigment, so instead of measuring absolute color, we were measuring relative loss of color. And this is what our data actually looks like. So what we're seeing here on this graph is that on the x-axis, we have our treatment. On our y-axis, we have loss in pigment inverted. So the lower the box plot, the more pigment was lost. And what you see are kind of 2 things really readily. Oh, and of course, within each treatment, each genotype is separated so that genotype 23 is blue and genotype 2 is in red. But you see two things very quickly. And the first one is that pigment loss increases with temperature, which makes sense. But we also find that there's a genotype effect where we're noticing that genotype 23 actually lost more pigment than genotype 2, illustrating a potential difference in thermal thresholds. And ANOVA did show that these findings were significant, and I'm running low on time, so if anyone wants to bring up treatment 9 here, ask me in the questions. Conclusions. So through this experiment, through this pilot experiment, we detected a possible differential genotypic response to thermal stress among two genotypes of Acropora Pulchra. There were some confounding variables. Size and the original, the size of the fragments were a little bit different. There was some paling originally with the corals before we put them into the experiment. Our findings do fit our environmental hypothesis. The coral that lost the least amount of pigment was the one found in the genotype that lost the least pigment was the one found in the southern lagoon, which could be some evidence of environmental priming, but we need more data before we can make any causal conclusions about that. But it's really cool because this shows that there are potential differences in our genotypes, and that can be used immediately in our restoration practices as we're going out and trying to put corals back onto our reefs and have a really healthy reef community. Next steps, we have a lot of things we're trying to do here: symbiont counts, lipid content, photosynthetic efficiency as other response variables to measure stress. If you have other questions, you can talk to me. And yeah, that is it. Thank you so much. I'm not sure if I went too over time for questions. Okay. Yeah, one in the back. Yeah, we have not started doing anything like that quite yet, but we are very interested in symbiont composition. We will be doing ITS2 testing so that we can determine the clade of the algae that is within each of these corals. It's already kind of well known that there are different clades out here that are more thermally tolerant than others, so that would be an interesting finding and more research to do. But we have not really looked at much of that kind of interaction between the different species and the different clades themselves. We also know that different species have very different levels of thermal tolerance as well. So, but I'm seeing my time's up.
So we do have one question from online. Ilan, in Photoshop, did you outline the corals by hand, or was there a tool?
So Photoshop has a pretty nifty AI tool that helps with that, but it tried to circle a lot of other things as well, so it didn't work out quite as well as I hoped. So in the end, we did have to do it by hand. And you would try to do it as closely as possible as you outline the fragments, but of course there's going to be some variation, but hopefully that's not, and as we continue these studies, we'll improve that as well.
Okay, thank you. And I think now we're going to go to our next presenter, which is going to be virtual, right? Mikayla Reuter is from University of Guam, and her discussion is going to be using CBASS to identify target species for coral restoration on Guam. So actually, I think your presentation was a really great segue into this. Thanks very much.
Yeah, I'm really excited about this one.
Hello. Can everyone hear me okay? Awesome. I see a couple thumbs up. Okay, let me try and share my screen really quick. Then, is that showing up as presenter view or as regular? Presenter view. Okay. How about now? Okay, perfect. Sweet. Awesome. And I'm just gonna start a timer really quick because now that I switched it over, I cannot see how long I've been talking for. Sorry. Okay, awesome. Thank you everyone for being here half a day. My name is Mikayla Reuter and I'm a graduate student at the University of Guam. I so wish I could be there. I really wanted to go, but unfortunately, I've been having some knee issues, so I'm back on the mainland for surgery really quick. But I'll be presenting a side project I worked on last year, which is also using CBASS to identify target species restoration. And during my talk today, I will be going over a quick introduction to talk about our study species and our study site. Then I'll go over the methods and the results of this study and discuss what we found, what that means for our restoration program, and what that might mean for future studies in Guam. So my study focuses mainly on staghorn corals, which are a subgroup of the Acropora family, and it builds on current restoration efforts in Guam that mainly also focus on staghorn corals. Restoration in Guam started in 2013 after our first major bleaching event there, and it focuses on these 8 species of staghorns, which are over here. Now, these staghorn species right now are identified mainly through morphological characteristics, so through how the coral looks, rather than phylogenetics or the coral's DNA. But we're working on getting some phylogenetic analysis in there as well. Staghorns are really ideal candidates for restoration because they're able to reproduce asexually, so you can break off just a small chunk of that coral and then grow it in a nursery setting. Staghorns also reproduce sexually, so we're able to keep that diversity up. Additionally, these staghorns have really fast growth rates compared to other coral species, so they can grow really quickly in nursery settings. And the fact that we have 8 species here on Guam allows us to restore a lot of diversity all at once. Unfortunately, these staghorns are also a lot more threatened by both local and global stressors. In Guam, local stressors can include overfishing or crown of thorns outbreaks or tourism and a lot of other things. But the main stressor to these staghorns is global stressors and especially bleaching. We've had 4 bleaching events on Guam now, and because of that, over 53% of the staghorn population has died off. Thankfully, we've already started restoration programs and outplanting around the island. And outplanting happens mainly at 4 locations right now, which you can see here. And outplanting happens at a range of different kind of environmental variables. So if you go down to Cocos Nursery down here, Cocos Nursery is a deeper lagoon. It doesn't have quite as much temperature fluctuation. And also there's a little bit less light because there's a lot of sedimentation in the water there. In comparison, up north here in Tuman, there's a lot less sedimentation, so the corals have more light, but there's a lot more tidal fluctuation, so the water can get a lot hotter, and also sometimes those corals are completely exposed due to the lack of water. Now, one thing that we haven't considered while doing these outplanting events yet is how interspecific differences in these coral species might impact outplanting success. And so what I wanted to do was ask the question, do staghorn Acropora corals on Guam have different responses to temperature stress? And if so, how might we implement that into our restoration program? And in order to do this, I use the same system that we just talked about in the last presentation, which is a CBASS system. CBASS stands for Coral Bleaching Automated Stress System, and it's a system invented by Chris Fulstra and Dan Barish. And they used it because there's a lot of locations around the world where it's really hard to do long-term heat stress. Long-term heat stress is useful in a lot of ways, but if you have power outages or if you have a lack of resources, then sometimes it's not really realistic to do these long-term heat stresses. On Guam, I actually finished this experiment just a couple of days before Typhoon Mara came in. So if I had been doing long-term heat stress experiments, I probably wouldn't have any data to show you here today. So personally, I am very thankful for the short-term heat stress systems. For this experiment, I went ahead and did a common garden experiment to try and minimize the amount of differences in the symbiont communities and the microbial communities between the different coral species. So all of our corals were collected from PD Bomb Hole Nursery, which is right here. And they were put into that nursery at least 4 months prior to the experiment happening, which there's been some preliminary research from UOG that shows that that's enough time to kind of minimize differences between those symbiont and microbial communities. For our different species, we collected either 10 colonies or 5 colonies from each species. And we did this 10 or 5 based on how large the parent population was. Every single day of the experiment, we collected our individuals that we were testing that day and took 4 coral nubbins from each individual and placed 1 nubbin into these 4 different tanks. And we had tanks at 29 degrees Celsius, 35, 38, and 41 degrees Celsius. 41 degrees Celsius might seem like a lot, but we did a couple preliminary experiments that showed that going to temperatures less than 41 degrees didn't quite make it so that the corals completely decreased in their fluorescence or their FVFM values. And that meant that we couldn't do the analysis we wanted to do. So we had to go up to that super high temperature in order to get the results. And as explained earlier, how CBASS works is your coral will slowly get stressed over this incline here as the system heats up to its maximum temperature over 3 hours. Then it's held at that temperature for 3 hours, and then it decreases over 1 hour. After that, we left our corals in the dark for at least 30 minutes before using a PAM fluorometer to test their fluorescence or their FVFM value. And what this PAM fluorometer does, it's a little machine that shoots kind of a laser light at the coral, and then it reads the signal that it gets back. And that tells us how stressed the coral is. So if that FVFM value is closer to 1, the coral's doing really good. It's pretty happy. If that FVFM value is low, closer to 0, the coral's not happy. It's probably dead. And usually around like 0.3 or 0.4 is where you see this coral start to bleach. And so what we got was for each individual coral colony, we were able to get an FVFM value at every single tank temperature, which you can see on this raw data graph here. And so each individual coral has a dot at each tank. And then from that, we were able to make a dose response curve for each individual coral. And that pretty much estimates at every given temperature, it estimates what the FVFM or the stress response would be. And from that, for all of our different species, we took every single individual dose response curve and we created a dose response model for that species to estimate what the FVFM would be at every single temperature. And then what we did here is we looked at a point at the inflection point here, and we figured out what the temperature is at the inflection point. And that's the effective dose 50 size. So pretty much that inflection point represents the point at which 50% of the fluorescence of the coral has decreased. So in more colloquial terms, it's the point where the coral is 50% on its way to being completely gone. And we went ahead and did that for every single coral species that we're restoring here. And you can see that not only do these ED50 values, these temperatures at which 50% of that fluorescence is gone, vary quite a bit, but also these gray areas, these confidence intervals, vary quite a bit. The reason we have a little bit of variation in these confidence intervals is twofold. So one of the reasons is because for some of these coral species, that ED50 value that is estimating falls in between 2 different temperature tanks. And when that ED50 value isn't close to either of the temperature tanks, the model just has to do more prediction. So it's a little bit less certain in that exact value. Additionally, as we just heard in our previous talk, there is some genotypic variation between individuals. So when a species had a little more variation in response to those heat stress tanks, then you might see a little bit of a larger confidence interval. Now, one thing I want to make clear is that these ED50 values are super useful, but they don't actually represent temperatures in the wild that these corals are going to stress at. Since this is a tank experiment and there's a lot of variables that are taken out that we have in the wild that we don't have in the tanks, and additionally, since it's so short term, we can't actually use these exact temperatures as estimators, but what we can do is we can use them to place corals into different categories and to compare these species to one another. So what this ED50 value does is it acts as a point where we can try and see what corals are more high resilient and what corals are more low resilient. And this graph does a really good job at showing that. So you can see here that Acropora virgata, acuminata, and vani are our low or medium low resilient corals. So every individual had ED50 values that were below 36 degrees for Acropora virgata, and then in that same kind of range for the other two. And then our high resilient coral was Acropora aspera especially. Now, since we can't use these exact temperatures, what does this really tell us? How are we using this data? Well, what we can do is we can look at these high resilient corals, so aspera, mercata, pteris, pulchra, and austera, and we can say that these high resilient corals would probably do better, have a higher chance of surviving if they were planted somewhere that has these high temperatures, so in a reef lap like Tumon. Whereas these lower tolerance corals probably wouldn't survive in Tumon, so when we're outplanting, we might want to only plant them in locations that have less temperature fluctuation, for example, a lagoon like Cocos down south. Now, there's a lot of other steps that we're going to want to do with this research. First off, I am going to be analyzing the phylogenomics and the population genomics of all of these corals. I have, I think, over 200, maybe close to 250 coral samples sent off to a sequencing center right now for low coverage whole genome sequencing. And we're hopefully going to be reestablishing those species boundaries based on that sequencing rather than just the morphometrics. And additionally, from that, we'll hopefully be able to analyze the symbiont communities and see if the Symbiotonaceae species and genre fluctuate between the different coral species. Once we know that, hopefully we'll be able to identify whether or not these stress responses are due more to the coral host themselves or more to the symbiont communities. Additionally, there's a couple future studies that might be beneficial. For one, we can add more populations to understand population level heat adaptation rather than just species level heat adaptation. We can also compare our species results or our CBASS results to in situ bleaching resilience. So does what we saw in the CBASS actually reflect what happens in a bleaching event? Or are there so many other factors happening in the environment that these short-term heat stress responses don't reflect this in Guam? And lastly, we can also go ahead and try and implement this technique of putting high resilient corals in locations that might have more heat fluctuation when outplanting and see if that actually works or if we don't see a correlation between success of outplanting and where we're putting these high resilient corals. Thank you so much to everyone who helped me with this project, my funding sources, University of Guam and the National Fish and Wildlife Foundation, my professors, the Island Evolution Lab, Remuda Coral Lab, and a bunch of other people at the Marine Lab. And now if you have any questions, I'm happy to answer them. And also, if you want to contact me, I left my email and also my YouTube channel where I put some science YouTube videos up there. And I so wish I could be there in person, but please contact me if you have any questions at all.
So we have one question here online. In your higher temperature treatments, did your corals start producing a lot of mucus, and how did you handle it? That's an interesting question. Awesome work, by the way.
Thank you. Yeah, our higher temperature treatment definitely had a lot of mucus. And I can say for one, as the one who siphoned out the water at the end of the day, I accidentally got the mucousy coral water in there, and I can confirm definitely very mucousy, so would not recommend. But in order to mitigate that, we had a pump that was pumping water through and a drip system, so water was constantly circling through. And then also we had a couple water pumps that were just moving and circulating water throughout, so that hopefully the mucus did not stay on those corals for too long.
Any other questions? That's a hard one to beat, I gotta say. Maybe you should move to trees. Hold on, let me just get to the back so that we can hear you a little better. Thank you. Go ahead, sir.
There was a nice graph there that showed the various tolerances of the various corals. Do you find that in the wild? When you look out in the lagoons, do you find that distribution?
Of where the corals are at depth?
You had a very nice graph about the corals and predicted where they might, or how much tolerance they might survive. Does that show up in the wild?
The high resilient corals are the ones that are found in the reef flats a lot of the time, but also the high resilient corals, since they can survive in both extreme temperatures and a little bit lower temperatures, they're found just about anywhere. But those ones that are low resilient, we find mostly in Opera Harbor or in the Cocos Lagoon. So those ones that don't do as well with heat resilience are really only in those deeper locations.
Thank you for your question. Anybody else? All right, thanks, Mikayla. We really appreciate your presentation.
Thank you so much.
And with that, we're actually going to have Lyza up next, and she's going to be talking about the development of novel settlement substancy of coral reef restoration using sexually propagated corals. Oh, are you going to talk about the Death Star? Yeah. All right. Yeah.
Can you hear me? Okay. All right, as promised, we're going to talk about our coral seed beads. These are novel settlement substrates, again, designed to improve the efficiency of coral restoration using sexually propagated corals. Okay, we already went over this. Okay, so coral restoration generally uses two approaches to propagate and create more corals for restoration. We use asexual propagation, in which a few fragments are collected from wild donor colonies, and then we grow and propagate those and continue to propagate those asexually in our nursery habitats. This is a really effective way to produce many corals from just a few fragments, and it has minimal impacts to wild donor colonies. They generally heal really well from these collections, and it doesn't phase them at all. You can also create gene banks in these nurseries to collect different wild genotypes and have those on hand to do things like thermal stress testing, selective breeding, and things like that. However, this method is very resource intensive. There is relatively little genetic diversity compared to sexual propagation, because each of these fragments that we take from a parent colony is genetically identical to the original colony. It's a lot like plants. You can, like, a lot of you are plant people, actually. That doesn't usually happen in my talks. But you can, take a little cutting from a plant and it'll grow into a new colony. But those plants are genetically identical, or at least I think so. Plants are very weird genetics. But anyways, so you have very limited genetic diversity. And again, this really limits the restored population's ability to adapt to any kind of changing environment in the future. And so we also try to incorporate sexual propagation. Again, we go out in the middle of the night and collect these eggs and sperm bundles. We help them out by fertilizing them and culturing the larvae. In the wild, very few of these gametes would actually result in a recruited coral to the reef. And so by collecting some of these, helping fertilization and taking care of the babies, we actually increase the number of new corals that are created from these spawning events. It also has the potential to greatly increase the restoration scale. Again, you're producing many more corals. It's a source of biomass and new coral genotypes for restoration that have higher genetic diversity and adaptive capacity again. So when corals are sexually produced, just like how humans are produced, each individual coral that results is genetically unique. So it greatly increases the genetic diversity of our restored corals. However, gamete collection and larval culture can be logistically challenging, especially at large scales. Again, these guys only spawn at night and only for a couple of nights over the summer. And if you miss that, if there's a storm that comes through or something, you're kind of screwed until the next year. Also, outplanting is still pretty inefficient, and survival of outplanted juveniles is low. And to try to address some of these challenges with sexual reproduction of corals, there's been a lot of work on developing substrates. These substrates were developed by our collaborators on this project, Seacor International. These are used all over the Caribbean to settle corals and outplant to restoration sites. They've done a lot of work with these and had some success. However, these are about the size of your fist. And if you can imagine, trying to outplant, tens of thousands of these over a large area of reef, it could get pretty unwieldy and logistically challenging. So we thought that maybe to go really large scale, we need to think smaller. Hopefully some of you are old enough to remember this movie. We decided to, take these sort of fist-sized substrates and shrink them down into about 1.5 to 2 centimeter substrates. And the idea was that these substrates can fit easily into a lot of these sort of nooks and crannies and grooves that are on coral reefs and that are actually more abundant now that a lot of the corals are dead. There's a lot of standing dead coral there with structure that these beads can fit in. All right, so these are the OG. I had someone not from the United States ask me what OG meant. That just means original gangster. I actually just made these with like some epoxy or something just to test out the size and how they might work. Luckily, we got some funding to further develop these. We got one of the Ruth Gates Coral Restoration Innovation Grants, and again, we partnered with Seacor International to further develop these substrates. And so there were a lot of ideas flowing around. We had a lot of different crazy designs that we were sketching out and modeling. But ultimately, after going through all of this, consulting with manufacturers, we decided on 3 initial designs. And we needed to call them something, so we have the Death Star, the Mini Gear, and the Gear Pod. Last but not least. And so again, these guys are pretty small compared to sort of traditional substrates that coral restoration practitioners have been using. They are made of 95% aluminum ceramic, which is a sort of next generation ceramic product that is non-toxic and should not hurt the environment at all. The next steps were to assess settlement rates of corals to see if they would even settle on them, if they like them. And then also to assess survival of the juveniles, both in the nursery and in the fields in various environmental conditions. And then also look at reef retention, so how well these actually stayed in those little nooks and crannies. So we started doing some of this. I'm going to talk about settlement rates and a little bit about nursery survival, and then we have another talk looking at more of our initial reef retention data, and that will be Sydney Madden coming up maybe right after me, soon. All right, so we first tested these guys with Acropora Pulchra. We collected the gametes in May of 2021. We had really good fertilization. We then put about 2,000 embryos in each of 12 replicate tanks or chrysals. We let them stay in there for about 4 days until they were ready to settle and metamorphose. We then took out any larvae that were left swimming around. And, We added 15 of each of the coral seed beads. We used to call them larval seed beads, so there's still some LSB stuff in there. But we put 15 of each of those designs and 3 Secor stars in each of the replicate tanks. We gave them another 4 days to settle, and then we removed any leftover larvae and counted all the settlers that were on the beads. Luckily, those guys fluoresce, so we can see them under a fluorescent microscope, even though they're very small. Okay, so these were sort of our selection preference results. Because these substrates, well, mainly the Secor star has a very different surface area than the other ones, we used Manley Selectivity Index to assess substrate preference, and this takes into account the proportion of available surface area and the proportion of settled larvae for each substrate type. So we did the math, and then this graph represents the data. We have means plus or minus the 95% confidence interval. This red line is what you would expect if they were just settling on them randomly based on their availability in the environment. And so if the 95% confidence interval overlaps with this line, they are not, they do not prefer that, or they're not avoiding it. They're just settling on it in proportion to its availability. So we found that interestingly, the larvae of Acropora Pulchra really preferred our gear pod design, which we're actually calling a football these days. These names are fluid. So they had a really strong preference for this design over the Secor star, and they seem to be settling on the other ones kind of indiscriminately. It's just some more of these data. We found, we also looked at, the percent of the LSBs that didn't have any settler, and this is kind of an important metric as we try to upscale this. We're going to be looking at just sort of subsets and outplanting large quantities. So again, the gear pod performed the best. Almost all of them had at least one settler, and they actually averaged over 20 settlers per bead, where the other 2 designs were lower. And this, of course, resulted in a much higher total number of juveniles produced on these beads. Okay. So we then sort of put them in these stainless steel mesh packets. This has evolved a lot since then. But then we put them out onto the reef, out into our coral nursery in the lagoon. And we let them sort of grow out for 5 months. We then brought them back into the lab and counted all of the survivors. Again, we found that the gear pod had the highest survivorship, but the mini gear wasn't too far behind. But because there were so many more of the settled larvae on the gear pod design, resulted in a lot more juveniles that had survived. And again, we have the number that didn't have any left, and the AirPod had the best performance. So that was interesting. We did some tests with 3 other species. Just due to a logistical issue, we didn't have the mini gear in this trial, but we saw the similar patterns. We had 2 more acroporid species, and they showed the same sort of preference patterns. And then we also had Goniastria retiformis, and they did not prefer any of the substrates. They were just settling on them in proportion to their availability. And we actually repeated these choice assays last summer, and we had all 4 substrates in there, and we had these species and 1 additional species. Unfortunately, I didn't have that data ready in time, but we're seeing pretty much the same kind of patterns. Again, more baby photos. These are just baby corals growing on the beads out on the reef. So far, we've only done some really small-scale pilot outplanting. Okay. So conclusions, the gear pod was most effective, seemingly small differences in shape. Microhabitat characteristics resulted in large difference in overall performance. Again, we're going to continue working on these, and you're going to see another talk in a minute on retention rates. And I'll leave it with that, and hopefully we'll have maybe a minute for questions.
I see a question in the back? Okay, I'll get to you in a minute, Steve.
Hi, that's really interesting. I just wanted to ask if you had any theory about why the gear pod design might have been preferred to the others, whether it has something to do with the topography, the combination of curved and flat surfaces, or anything like that?
Yeah, that's a great question. I think it probably does have to do with the topography. Again, that had a smooth or like a kind of a curved structure. You know, the Death Star was also round, obviously, but we designed that one to have the corals actually settle on the inside so they'd be more protected. Because we do know that coral larvae like sort of settling in these like little ridges and nooks and crannies. And so, yeah, exactly. I think it's probably a combination of having both of those microhabitat characteristics. You know, whether, you know, if we think about why they would prefer a curved surface, it might have something to do with sediment sloughing out on the reef, where sediments might, you know, flow over curved surfaces more readily than a flat surface. But that's something we're still investigating. But yeah, it was a very interesting finding. Thank you.
Thank you.
All right, and I know we have a question from Steve. If you could meet me halfway, that would be great, but no, I'll run over. It's okay. I got it.
Hey, Lyza, you guys are doing awesome work. It's really incredible the amount of science you guys are adding out here in this remote place, obviously. Seeing what's been happening in the Atlantic Caribbean over the last few months, last year, with their massive losses and failing restoration efforts, despite all of the science and all of the history they have there, are you seeing lessons from what's happened there that we can apply here? Do you see a way for us to move forward where we're not going to run into that same catastrophic result? Because I feel like we kind of have been trailing behind them by a few years, and what we don't want is a few years from now for us to hit that exact same hurdle that they just hit, right?
Yeah, absolutely. That's a great question. You know, we are trying to take some steps to avoid that issue. As I mentioned and as Ilan talked about, we are trying to improve the thermal tolerance of our outplanted corals. That is something that they didn't really do in the beginning in the Caribbean. They really just relatively recently, before this big bleaching event, started, you know, thinking about that and trying to selectively propagate thermally tolerant corals. So that is definitely a lesson that is learned from that situation. Again, also increasing the genetic diversity is a huge part of making sure that these reefs are more resilient and can respond to the changing environment. Luckily, and you know, our reefs, the Western Pacific reefs, are a little more inherently resilient. We have much higher coral diversity and functional diversity of corals out here. You know, historically, we haven't seen the huge disease outbreaks and the other issues that they have in the Caribbean, where they have, you know, only something like 36 species of coral, and we have over 300. So the ecosystems have a little bit of more resilience built in. There's also some research out of Palau that's showing that those reefs have, you know, are adapting somewhat to the warming water, and that there's corals seem to be showing an increase in thermal tolerance through time. So there's a little hope there too. So yeah, we are definitely, everything we are doing is focused on increasing the resilience of our individual corals that we outplant and the reef ecosystem as a whole.
Really appreciate your time and your efforts. And I know Sydney Ballard is up next, right?
Hello, everyone. My name is Sydney Madden. I am a research associate with Johnston Applied Marine Sciences. And today I will be telling you the next step of what Lyza was just telling you, talking about reef retention rates of novel coral larval settlement substrates, you already know as CSBs, on exposed forereefs of Saipan. As we now know, and already know, coral reefs are crucial ecosystems. They provide many ecosystem services globally and locally. Globally, they are biodiversity hotspots. They are also, they act as a carbon sink. And locally, they are being used as a commercial and recreational fishery. They provide coastal protection. They hold a strong cultural, religious belief and spiritual importance. And they are a draw for tourism. There are many challenges of for coral in the CNMI, from thermal stress and bleaching to land sources of pollution, natural disasters such as typhoons, and other human uses. Unfortunately, due to these localized factors, a large portion of reef structure has been lost. So, therefore, coral conservation is no longer focusing solely on preservation practices, but primarily coral restoration in the forms of asexual and sexual propagation. Sorry, we're going to go over this again. Asexual propagation is also known as fragmentation, and is when coral is cut or broken into pieces, with permits, of course. Supportive breeding through sexual propagation is a strategy that has the potential to increase coral cover and introduce novel genetic diversity, improving the resilience and adaptive capacity. There's also a large reproductive output, which is a great source of potential coral biomass. So CSVs, they were designed in collaboration between JAMS and Seacorps International in hopes of increasing the output of sexually propagated corals on reefs, which is a limiting factor in coral restoration. These small ceramic substrates, the ones here in the picture, the footballs as I like to call them, they fit in the palm of our hand. And how do we get larvae or coral juveniles onto the beads? So first we have to go out during coral spawning during the lunar cycle, which follows a lunar cycle in summer months. We went and collected gametes from parent colonies. We then fertilized them and put them into our land-based systems, which is the second picture here on the left, and our oceanic systems as well. We have a closed contained system so the coral larvae won't float away. For about 7 days, the larvae are free swimming until they decide to swim down and look for bare, hard substrate to settle upon. These coral larvae look for settlement cues like CCA or coralline crustose algae. The beads that we have in our systems and put in the systems, they've previously been conditioned out at our oceanic nursery to entice corals to settle on them. The beads are also designed with many groups for preferred settlement as they like nooks and crannies. The ridges also provide protection from physical abrasion and predation and will hopefully help the beads self-stabilize once they are outplanted onto the reef. These beads are also efficient for outplanting for many reasons, everything from being timely for time purposes. So instead of using such practices for outplanting, such as using zip ties or epoxy, which is like a putty-like substance, or cement, which can be really messy, you just have a diver or a snorkeler and you just put it and you kind of try and wedge it into a part of the reef where it won't float away or move with wave exposure. And they're also very cost-effective. Our project objectives can be broken down into 3 pieces. We've talked about our design substrates. We now have 5 going on 6. And we tested for CSBs for preferred larval settlement in Lyza's presentation. And now we are looking at CSB retention and juvenile survival in this presentation on the forereef. A different study had a goal of testing CSB resilience to assess juvenile survival across a reef resilience gradient, which takes into a multiple of factors. The results I will be showing you today are only preliminary, as we have 2 data points thus far. So when looking at the retention of CSBs in this study, we will be We're looking at how many beads stay within the designated plot area as well as generally around. Another data set we looked at were how many beads remain stationary within the plots between the two time points. We assume that CSBs that stay stationary will do better due to the biology of corals and how we place the corals, our methodology. So on the biology of corals side, corals, they can't exactly grow over the substrate if they're being rocked and rolled around on either hard substrate or in the sediment. So with our methodology, we try and place our corals away from other corals that are growing on the reef, other algae, so they aren't smothered, and away from the sediment so they aren't being smothered in that way either. And in this picture, as Lyza already said, you can see where we had outplanted those beads on the reef. The original goal was to test the CSB survival across reef resilience gradient by looking at these three sites, Lanius, Elbow, and Mandy, with different resilience values. Lanius had a high resilience value, Mandy had a medium high value, and Elbow had a medium low. The reef resilience gradient was determined from a reef resilience assessment conducted in 2019. However, due to low settlement of corals during that specific spawning season that year, we are unlikely to see survival signals, so we are simply focusing on retention. Although we will assess survival at the next monitoring points from here on out when we go back to these three sites. And we are also testing retention and survival at multiple other sites. So more on the methodology side of things, each site has 10 replicate 1 meter radial plots that were established along a 7 meter plot gradient or depth contour along a 100 meter transect. 25 beads were placed into grooves or other structures in the reef by a diver. Then the plots were monitored at the time of outplanting and after a 4-month period using structure for motion photogrammetry. So what is structure for motion photogrammetry? It is a method of monitoring restoration and outplanting sites. In this time, we take hundreds of overlapping high-resolution photos of each time point and plot. We use a computer program called Agisoft Metashape to compile the photos to create a 3D model and an orthomosaic, which is like a really big picture of everything put together. So this graphic shows a 3D model of one of our August time points. As the bees were just outplanted in this plot, we can more readily see them. In this section up here, you might see some white specks. There's one right here. That's not it, that's just a marker. So there are lots of benefits to using structure for motion photogrammetry, as it is not as time consuming in the field. So this is the same plot just 4 months later in December. So while there are benefits, such as it's not being as time-consuming in the field, you swim over, you take pictures, you might get a little cold because you're not moving that much, but the big disadvantage is the processing time on the back end. It can take upwards of 8 hours to create one of these plots, sometimes more, but it all depends on your computer processing power, GPU, CPU, some other acronyms I still don't understand. But this can be mitigated by getting better software, and it depends on where you are. Being on an island in the middle of the Pacific, it might be a little more difficult to get those types of software and things. However, if you have a great coworker who can build one for you, it's even better. So a few other benefits is that you have this long-term photographic record for reassessment, and you can measure other parameters post-survey, like we will use these models to measure rugosity, which will be another point in this study. So this graphic is an orthomosaic, which is an output of Agisoft, that really big picture compiling hundreds of photos. So this shows change of CSBs over a 4-month time frame, their position over that 4-month time frame. The green dot shows the position of CSBs in August, and the pink dot with the white halo around it shows the position of December CSBs. So if you see a halo with a green dot in the middle, it shows that those are the beads that remain stationary. So in this plot, this is on a December orthomosaic. In the August plot, there were 25 CSBs that were originally outplanted and found. Sometimes they kind of get lost. And then in December, we found 15 of them within this plot, and 5 of those 15 were stationary. So we have 2 stationary ones down here, 1 to the left, and 2 up there. You have to look really closely at those. So this graph shows the percent of CSBs that were retained between the 3 sites. The mean plot retention ranged from 22 to 43 beads per site, with an average of 32%. There was a significant difference between these 3 sites. These are preliminary results, so we didn't do any further tests just yet, but it's interesting to see that there may be a difference between sites. We will investigate further with more data and taking into account other factors. This graph shows the percentage of CSBs that were stationary across the 3 sites. The mean stationary bead percentage ranged from 10 to 19 beads per site with an average of 15%, and we did not find anything that was statistically significant. So these results showed a higher rate of retention on exposed forereef than expected. It was expected that retention might be lower than 32% because they might be blown away by wave exposure, and especially compared to more protected areas like the lagoon or the back reef. However, there was somewhat higher retention rates than expected at Mandy, so this could be due to less wave exposure, kind of being in that elbow of the north part of Saipan, and we'll be looking into that further. Going forward from here, we will gather more data points to see how these CSBs have moved in a year time frame, maybe an additional one from there too. We will look at structural complexity and wave exposure on bead retention to see if there's a correlation there. And right now we are doing an active retention study. I always get this mixed up. Retention study in the lagoon, looking at varying deployment methods. Right now we're doing the placement method. We're doing the placement methods, and we will be, we're looking at sprinkling, which will decrease the time even more, so you can honestly be on a kayak swimming or kayaking around and then just dump the bees off the side versus having someone have to get in the water and manually place every single one. So more to come on that side. So I want to have a big thank you, say a big thank you to Lyza, the JAMS team, and everyone on this list, doesn't even include everyone, for their contributions towards this project and presentation. Thank you for your attention, and I will answer any questions you may have at this time.
So do we have any questions so far? I actually had a question because you had mentioned that CSBs are cost effective, and so and I know that Lyza talked a little bit about the manufacturing of these CSBs and just being in an area that's kind of hard to send things to, right? Can you discuss a little bit how much these cost, maybe relative to the other ones that C-Corps had developed, like the big star ones? I would be really interested to know that.
While I do not know the specifics of that, I believe that each CSB costs less than $1, and then even in some of our new designs that we have made, we can make them ourselves with making cement and making our own molds. So that even
awesome.
Brings down the cost even more.
Okay, that's great. And I'm sorry, what's the actual material that you're making them with?
Ceramic.
Okay.
I believe it's 95% something ceramic. It's a fancy type of new ceramic.
Did anybody else have any questions? Okay, well, the good thing is that we're going to be coming up into a break, so if you have any, oh, we're not? Okay, I guess we are not. Oh, yeah, that's right, because the poster session is next, right? Okay, all right. Thank you very much, Sydney. Thank you to everybody on the restoration panel. It's okay. Yeah. Thanks to everybody on the restoration panel, and Anne-Marie has a couple of announcements.
Yeah, thanks everybody. We're getting towards the end of our day here. Just a few quick announcements. I know there is a break on the schedule, but you're going to be able to very soon walk around the room and look at the posters. So it'll be like a break, and there will be refreshments arriving very soon. So just a few announcements. If you are a potential mentee, please sign up for our mentor session. Lunch will be provided tomorrow. We also have our job fair tomorrow evening in this room. There's also still more room on almost all of the field trips, except for Luta and the Omelet Walk. So please sign up for that. If you need help with signing up for any of these things, just approach one of us and we can help you through that process. Also, we have a hashtag. So if you want to post about this conference, it's MICC Saipan 2024. And maybe we'll actually put that on a slide tomorrow. So we're going to do something with the posters that's a little bit different than you might be used to. We did this last year and it worked really, really well. So can I have all of the poster presenters please come up to the front? They were asked to submit just a slide, kind of either summarizing their poster or of their poster, and they're going to give maybe just like a 30 minutes, 30-second spiel on their poster. So can we have all the poster presenters please go ahead and come up to the front? To the stage. So this is going to be like shotgun. If you see your poster, if you see your slide appear, come up and give your spiel. Or go ahead and introduce yourself and give your spiel. You can go ahead and come up on the stage, yeah. Oh, everybody. Thanks. Right, what a great group. And I'll just, make sure we can see the slide. So maybe come over to the sides. And we'll have that coming up in just a second. Waiting for it to download. Hey everybody, stop clogging up the internet. All right. What you didn't know is we're going to make you all dance now. Still loading? All right. Oh, nice. Oh, looks like we're going to hook right into Haldra's laptop. Yeah, and I think one of the great things about this format is it's a little bit better for folks who are attending online. They can actually be a part of the poster session. Oh, I guess I can take this time to thank our anonymous student talk and poster judges. They'll be going around to the posters, and we've got volunteers for each day to judge the student talks. There's a couple of student talks every day, and we'll be awarding them prizes on the last day. So Kaylee just had a special request. Could we get a show of hands for who is interested in trivia? This is going to be tomorrow night. Trivia, I'm raising my hand. At Mariana's Creations, that's at 7-ish, right? Yeah, 7. Interested. Okay. Thank you.
Hello, everyone. Are you aware of CNMI food waste management methods? If not, you fit into the 92.6% of CNMI residents who were unaware of food waste management methods. This reveals the large gap in knowledge about food waste and how to properly handle it. Global food waste is a critical challenge with far-reaching consequences. Our research aims to enhance sustainability within the local community by addressing this environmental concern. Thus, we want to capture biogas from food waste and utilize it as a renewable energy source. Do you want to learn more? Come and visit our poster presentation about CNMI Renewable Energy Initiative. Thank you.
Thank you. Oh, yeah. And where is your poster located? All right. So if you could point in the general area of your poster. Yep. Next, we've got V.
Half a day, everyone. So I have a poster which is just on the back in the green. So it's a poster that gives you an overview of biosecurity inspections that we do with SEMOL, all of the hard work detecting live materials, live and dead biomaterials, to avoid any spread of invasive species, and that's rundown for fiscal year 2022 and 2023. And that poster was made with Stephen Mosher that couldn't come today, but I will be talking on his behalf. Thank you very much, and I will see you out there.
Thank you.
Gwelu zengwela kusinya malofangitano mizu. How do people listen? And how does sound reorient frameworks of perception? So my research traverses the archipelago by way of acoustic environments and the community's interventions in such environments, and how we come together with our voices, within sound, within non-human beings. And I investigate the conditions that make them possible or impossible. So it situates indigenous song, dance, and the work of cultural practitioners as crucial in order to better understand habitats in duress or that are no longer here. So this is a kind of knowledge in my project called Sound Knowledge that can only be experienced in sound, and it expands possibilities of environmental stewardship. For example, what you see here is me at the Tonkununu, and in the Chamorro context, we might view this as an ancestral absence made present via the Tonkununu, an interplay of culture, a human of life and death. And so we communicate this with our voices and acknowledge such presences. And so This is so what I'm looking at. My poster's over there, it's in the blue, and so you see many practices in Saipan, from Talabok to the central part of Saipan, and so my project is to sort of gather these sort of knowledges via sound, via these practices, and sort of to investigate what type of knowledge this can bring in towards many disciplines. And so I'm glad to be here in this conservation conference. Sizus Masit.
Hi, my name is Rachel from the San Diego Zoo Wildlife Association, and my poster is discussing some of the successes of the AGA captive rear and release program on Rota. So it essentially just goes over some of the techniques and methods we use for hand rearing and raising the crows in our care, and some of the ways we help prepare them for release, as well as some of the successes we've seen after birds have been in the wild for a little bit.
And where's your poster? Over there. Thank you.
Hi everyone, my name is Eunice Molina, and currently I am a junior here at Marianas High School. And last summer I worked with the Ecology of Birdloss project, where we observed pollinator visitation to flowers, specifically in Saipan's limestone forest. And so what my poster will be talking about is ways to improve camera trap techniques for more accurate observations. And my poster is back there in green.
Thank you. Project Hope.
Good afternoon, everyone. My name is Alex, and this is Kina. We're here on behalf of Friends of the Mariana Trench to showcase our program, Project Hope.
Project Hope is a collaborative ocean science club amongst PSS students, college students, outreach specialists, and community elders. Project Hope's main initiative was to increase ocean science aptitude and stewardship amongst middle school students by combining Western science and traditional ecological knowledge. If you're interested in exploring the effects of Project Hope on our community students, we invite you to check out our poster right there by the refreshments. Thank you.
Mark?
Here we go.
Hafedezen tero manyeluze manenao gawin anu si Maktalai Pangilinan Junior giz Astalai Hills and Northern Mariana College. Now, one of the beautiful things about the CNMI is that we have many biodiverse ecosystems, from our coral reefs all the way to our limestone forests. Everyone knows about our tropical birds like the totot, palumanapa, the titirika, nahabak, and everyone knows about our local trees as well, tronconunu, tronconumumu, tronconpagu. Well, what most people don't know is that we also have a wider range of ant species on the island. Now, good news is we have more than, say, like 40 or 50 ant species on the island. The bad news is that also places at great risk of dealing with invasive species. This one in particular that I've been studying for about 8 months is Anapolepis gracilis, better known as the yellow crazy ant. If you want to know more about their geographic distribution, biology, and behavior amongst our native species here in Marianas, you can find my poster over there behind the camera. And I also have some yellow crazy ant specimens pinned up in a box as well for closer inspection. So it's Masis and Gilisa.
Hi, my name is Rebecca. I'm a wildlife biologist stationed on Tinian with the CNMI Division of Fish and Wildlife. We're doing a cooperative agreement with NAVFAC Marianas, studying the long-term monitoring of the Mariana common moorhen, Gallinula chloropus guami. If you would like to come talk about their seasonal distribution, seasonal distribution and population trends over time, my poster is directly back from here.
Thank you. Is Frank here? Maybe his poster's. Let's go ahead and move to the next one. okay. There you go.
So the last poster in the session is from E.J. Aguirre and co-authors, and it is, what he wanted me to read was the abstract, which says, studies have shown that art therapy is an effective treatment for the remediation of mood and anxiety disorders. The current study explored the potential therapeutic benefits of reenacting native cultural customs, emphasizing crafts and performances as forms of emotional therapy. The authors assert that these traditional activities, being essential to a person's indigenous identity, promote healing. Examining 4 creative art therapies, art, dance, music, and a combination thereof, we found that traditional crafts and performances, such as canoe building, dance, and storytelling, significantly enhance emotional well-being by fostering a profound connection to one's culture. And he is a student at Northern Mariana College, and I am not sure where the poster is, but over here, it's right over here. All right.
Thank you. Can we go back to Frank's? No worries. Can we go back to Frank's?
Half a day, everybody. So sorry. I misjudged the schedule. So my name is Frank Villagomez. Sorry, let me catch my breath. I just had to. I just literally just came from the parking lot. My poster's back there in the back corner. I'm gonna be talking to you guys about the age-based life history of the pink snapper, Opakapaka, as it's commonly known, Priscipomortes filamentosus. Basically, this is some of this study is what I did on my master's at University of Guam. So, yeah, I'll be back there, and I'll be happy to speak with you.
Thanks, Cole. Is that it? All right, let's give just one more round of applause for all the poster presenters and for a successful, almost finished first day. Okay, you're free to roam now. Yes, there will be snacks soon. And if you want more drinks, you can go buy them at the lobby bar.